So thank you for coming here today. It's truly a great pleasure for me to introduce to you Thomas Mcdade. He's a good friend of the center and the demography program. Thom is the Carlos Montezuma Professor in the Department of Anthropology and also a faculty fellow at the Institute for Policy Research at Northwestern University. Thom is a biological anthropologist specializing in human population biology. His work is primarily concerned with the dynamic interrelationships among society, biology, and health over the life course, with emphasis on life course and approaches to stress and the human immune system. He has done considerable international work, including in Samoa, Bolivia, and Ecuador. Thom is also the director of the Laboratory for Human Biology Research and director of Cells to Society, Center for Social Disparities and Health. His work has been supported by grants from the NSF, NIH, the Winner Grant Foundation. In 2002, he was the recipient of the Presidential Early Career Award for Scientists and Engineers. Thom is a highly accomplished researcher. He's an elected member of the National Academy of Sciences and of the American Academy of Arts and Sciences. So it's truly a pleasure to introduce to you Thomas Mcdade.
I think this is on. Can you hear me? Great. Okay. Well, thank you all for having me. It's been a long time, but long time that I've been here under the auspices of PSC, but I have a lot of great friends and colleagues, collaborators here, so it's always fun to come back. And I was saying to Don, my mom grew up in South Philly in Essington, so I spent a lot of time in Philly growing up, but it's been a long time since I've been back, so I thank you for the invitation to return. All right, so I'm going to talk to you today about inflammation. And so my training's in biological anthropology and population health, and I'm primarily overarchingly interested in biosocial life course approaches to population health. This talk is going to have a little more bio than social, and it's because I've been doing a lot of work on inflammation over the life course, and most of my work focuses more on the earlier side of the life course, but inflammation has generated a lot of interest in the context of aging. And so I want to share with you some thoughts about what I see as three common assumptions guiding current research on inflammation, aging, and health that I think are probably wrong, or at least worthy of some critical inspection. And so that's what I want to do with you today. So the first assumption is that everything we need to know about inflammation can be learned from research in affluent industrialized settings. The second is that studies of aging and health begin when people turn 50 or should do so. And then the third is inflammation is synonymous with pathology. So I'm going to break each of these down. Let me say I welcome questions along the way, and there should be time at the end as well for Q&A. But if something comes up in the meantime, please let me know. All right, so let's start with why we should care about inflammation. Inflammation is a key part of our innate immune defenses against infectious agents in particular. And if you do not have a functioning inflammatory system when you are born, you don't make it out of infancy. It's just that critical as a frontline defense against ubiquitous pathogens that are a part of our world. But in addition, inflammation plays key roles in wound healing and tissue repair. And also, I think a lot of people don't appreciate just how important inflammation is to basic physiological processes related to growth and development. For example, brain development, brain structure is a process of proliferation and connection of neurons and axons, and inflammatory processes are critical for pruning synapses.
It's right on the computer where the.
Hello?
Yeah. Is the sound on or no?
What? Who's that?
Maybe someone on Zoom.
Apologies, it's muted.
Okay.
Okay, very good. All right. So I think people don't often appreciate the important roles that inflammation plays in basic processes related to brain development, structure, and function, as well as bone remodeling in the context of child growth in particular. So it's a pretty important basic inflammatory or physiological system. But there's also a darker side to inflammation. And over the past at least 20 years, we've come to appreciate how inflammation can contribute to cardiovascular disease, diabetes, Alzheimer's. 2004, Time magazine labeled inflammation the secret killer. And of course, we have a lot of prospective cohort studies, most of which begin around age 50, gather baseline assessments of inflammation and other markers of cardiovascular risk and follow people up over time. This is a meta-analysis which shows that the risk ratio for all-cause mortality prospectively with a baseline C-reactive protein measure, which I'll explain more in a minute. There's a significant increase in risk in all-cause mortality, and we'll especially see this in the context of cardiovascular disease mortality, more than double the risk if you have high inflammation versus low inflammation in adulthood. So CRP is a endpoint biomarker of inflammation. It's produced by the liver. It's relatively easy to measure in blood sample, relatively inexpensive, and it's widely used now clinically. Last time you had a physical and a blood draw, in addition to your lipids, they probably tested your C-reactive protein. So it's being widely used clinically as well as epidemiologically to assess baseline levels of chronic inflammation. All right. Now, it's also apparent that chronic or dysregulated inflammation has implications for health across the life course, not just in the context of aging. So we have earlier in the life course, prenatally and early postnatally, when brain development is happening most quickly, dysregulated inflammation in utero or in the fetus or the baby can have implications for brain structure and ultimately function, leading to autism or schizophrenia. In childhood, inflammation is involved in the etiology of asthma and allergy, which affects obviously a lot of kids and adolescents. It's also involved in depression. It's also a cause and a consequence of depression, and dysregulated or high levels of inflammation can contribute to preeclampsia, preterm delivery, and other maternal morbidities and adverse birth outcomes. And of course, for our perspective as interest in population health, exposures like psychosocial stressors, excess adipose tissue, toxic exposures, smoke, environmental contamination, smoking, latent infections, these can all lead to elevated levels of chronic inflammation, and they're socially patterned. So for these reasons, inflammation has been a major focus of health inequity research and biodemography in population health. So that's why we care about inflammation. But is everything we need to know about inflammation, can that be gained from research in industrialized affluent settings? So first I want to tell you what the model is that comes out of research from these settings, and I'm calling it here the chronic low-grade inflammation model. So this is a study that was done 25 years ago, and it's pretty foundational, widely cited. And what they did is they took blood from 4 people and drew it every 4 days across this 2-month period, and just plotted the concentrations of CRP. And what you can see is that in general, things bounce around a little bit, but in general, people are relatively stable. You can identify the people who are consistently low in their CRP and the people who are consistently high. And you can see it a little bit more clearly here, where it's the same study, but monthly blood samples over a 6-month interval. And these are the participants plotted here on the x-axis, and the y is their CRP. And then here, 3 milligrams per liter is widely used clinically as a threshold for differentiating high-risk CRP from lower risk. And you see individuals tend to cluster. So there are people who are consistently above this high-risk threshold and people who are consistently below, and some who are kind of around the middle. And so the authors of this study conclude, based on these sort of biovariability analyses, that their data suggests over a 6-month period, CRP values are relatively tightly regulated, with some individuals having consistently higher values than others. So the model is that there's relatively high levels of between individual variability, relatively low levels of within individual variability. So we can measure CRP once and get some level of chronic baseline level of activity. That's the assumption validated by these kinds of studies. So what does this mean in terms of sort of a broader population health perspective? When we measure CRP in big demographic surveys, or in this case, NHANES, we see that in general in the United States, CRP tends to go up with age. And then most studies report a sex difference with women tending to have higher levels of CRP than men. And then if we look at patterns with overweight and obesity, which is among the strongest predictors of chronic inflammation. We see in the United States, these are AdHealth data, wave 5. Yeah, Amelia.
Yeah, no, that finding that you have people, above or below or something,
Yeah.
is it connected to behavioral, to any social, or is just biology that you get, that division?
Right, whether someone would be higher or lower
Higher or lower,
consistently.
yeah.
All those things. So chronic inflammation moves, especially in relation to excess body fat. So if you have visceral central body fat, that's a real driver of chronic inflammation. So if people have higher BMI or waist circumference, it's showing here for males and females, have a significantly higher risk of high CRP. So only about 10% of people at normal weight are in that high CRP group, but as weight goes up, probability of high risk CRP goes up. Stress increases chronic inflammation, smoking increases chronic inflammation. There's a genetic contribution as well. All right, so these data are from Wave 5 of Ad Health. And then this is to make the point, what might we be missing if we construct our understanding of inflammation, how it matters to our health, only by research in places like the US. And so this is a figure from Gapminder, which is probably many of you are familiar with, a great source of sort of global health statistics. And what I've plotted here on the y-axis is energy balance, is proxied by body mass index. Each circle represents a country. The size of the circle is proportionate to the population size of the country, and the color denote different regions. And then on the x-axis here, I've plotted infectious exposures, low to high. So just what's epidemiologic, what's sort of the overall infectious disease environment of a particular country, and I've proxied that very simply by diarrheal deaths in children before age 5. And this is the US. So we are a low infected, overfed quadrant. And then, but that's not representative of how most people are in the world, right? So most people are down here, high rates of infectious disease, relatively lean. And I'm going to talk about 2 case studies in my own research from the Philippines in this quadrant, and in Ecuador, high levels of infectious disease, but slightly higher average BMI. All right, so I think, as an anthropologist, one of my responsibilities is to challenge the assumptions of universality and challenge the definitions of normality that are based exclusively on studies conducted by people living in affluent industrialized settings. So let's go to the Philippines. This is the Cebu Longitudinal Health and Nutrition Survey, which began in 1983 and 1984 with a recruitment of 3,327 pregnant women, representative of childbearing age in the area of Cebu metro area, which is the second biggest metropolitan area in the Philippines behind Manila. They gave birth to their babies. The babies and the moms have been followed up ever since. And we have major survey waves in 2005 where we collected blood samples, which I'm going to show you some results from. We've now sort of transitioned to funding from NIA, from NICHD, because we're pitching this as an aging cohort. So we have a new round of data with the moms, as well as the Gen 2, the original cohort members who are now in their early 40s. And I did a postdoc at UNC Chapel Hill in 1999, 2000, and that began my affiliation with the Cebu Longitudinal Health and Nutrition Study. All right. So this is what CRP looks like in the Philippines. So just for comparison, this is NHANES data, age matched. So the participants that we're looking at now are young adults in their early 20s. And in the United States, overall, the median CRP is 0.9, a little bit higher for females than for males. In the Philippines, in our cohort, it was 0.2, 0.3 milligrams per liter. So much lower average concentration of CRP. And this was initially a little head scratching for us, because we thought, well, this is a higher infectious disease environment, maybe there'd be more inflammation. In fact, much lower level of chronic inflammation. So one possibility is that this is just adiposity. So I showed you earlier that body fat drives chronic inflammation. Filipinos tend to be leaner than your average American 20-year-old. But if we stratify by waist circumference, which is a stronger predictor of inflammation than BMI even, you see that even within each stratum, there is a higher level of inflammation in the United States compared to Philippines. And it's not just the Philippines. So you might say there's something genetic, unique about that population, or something dietary. If we do the same comparison in older adults, 18 to 50-year-olds in Ecuador, in an area which I'm going to explain a little bit more about the subsistence and ecology of that group momentarily, but we see the same thing. At the same level of adiposity, much lower levels of chronic inflammation in Ecuador compared to the United States. And then if we look at the association between inflammation and age in our moms in the cohort in the Philippines. So at this survey in 2012, the moms range from mid-40s to mid-70s. And we just a simple plot of age in relation to log CRP, and it's totally flat. So we don't see that upward trajectory with age that we typically see in the US and other Western affluent countries. All right, so what's going on here? When we broaden out a measure of inflammation outside of industrialized settings, we're getting some paradoxical results, and they encourage us to ask whether we can assume that this chronic low-grade inflammation model shown here is actually universal. Right? So to answer that question, we conducted the first study of CRP variability in a high infectious disease environment. So now we're going back to Ecuador, and we're working with the Shuar, who are an indigenous group of horticulturalists, live in small clustered villages and basically subsist off the land with some small scale farming and a little bit of trading. For our purposes here, fortunately, the rate of infectious disease exposure and death is quite high, 5 to 6 times higher than it is for a child in the United States. And the main sources of morbidity are acute respiratory infections, gastrointestinal illnesses, and vector-borne diseases. So we implemented a study with 52 adults, 18 to 15 years old, with 4 sampling intervals. Very simple study where we collected finger stick dry blood spot samples. So as you can imagine, the logistics and the infrastructure here did make it challenging to collect blood samples and bring them back to the lab. But we use dry blood spots in my lab, and much of my research is a way to get blood in, for basically $1, we can get a blood sample from just about anyone, anywhere in the world, and bring it back to the lab where we can quantify it for C-reactive protein using amino acid that we validated a long time ago. And now before I show you the results, I just wanted to prime your memory with, this is what the results look like in the United States. High levels of between individual variation, very low, relatively low levels of within individual variation. And this is what we get in Ecuador. So these are participants. Participant numbers distorted on their sort of average CRP. Each diamond represents one of those 4 CRP observations. And then this 3 milligram per liter line is that sort of high level, high risk CRP couple. There are no people who have more than 1 elevated CRP measure, right? So there are no clusters. The people who have a CRP elevated above 3 also have observations below 3. And the pattern is a little clearer if I just separate out these people, about a third of the sample that at any one point in time over the course of the study had a high CRP. So now I'm plotting these 4 weekly sampling intervals. And what you can see, if you look at interval 2, there are a handful of people who have high CRP. But at the next week, they all have CRPs that are approaching 0. They're below the threshold. And then at week 4, there are a bunch of people who have high CRP, but they're different than the people at week 2 or week 3, right? So by measuring CRP in the same sample of Shuar adults across 4 weekly time points, we have multiple cases of elevated inflammation, but every time CRP went up, it also came down. There are no people with consistently high CRP and not a single case of chronic inflammation using consensus guidelines for identifying chronic inflammation in the United States. So the practice, the pattern here is effectively opposite what we see in the US. It's high levels of within individual variation and low levels of between individual variation over time. All right, so what are we missing if we construct our understanding of inflammation based exclusively on research in people in the United States? First, we see that CRP is relatively high in the US, at least at a chronic level, and it's more strongly associated with adiposity at age. But we can't assume that that's normal or universal, right? Inflammation is chronic in the US, but not necessarily so in these other epidemiologic contexts. And if we take a broader view, do research on inflammation in different ecological settings, maybe we'll get some just on why there are these different patterns of inflammation across these settings. All right, next assumption. Studies of aging and health should begin when people turn 50. Hans Peter isn't here. How many people are from the aging center here? How much, is there a lot of overlap between the aging center and PRC? Yeah.
Well, you have a bunch of old people over 50, I can tell
That's true. Yeah. People, I don't want to offend scholars, but NIA has a website with their surveys of aging supported by behavioral and social sciences, and they all begin when people turn 50. Right, and some creep back a little bit. And again, many of these studies try to reach back and get some information on experiences and exposures early in life. But for the most part, the assumption is if you're going to do a study of aging, you begin it in middle age and then go forward. And I think that's fine for many questions. But there's reason to think that that might not be sufficient for understanding how inflammation functions and how it relates to diseases of aging in particular. So most of you are probably familiar with the developmental origins of health and disease, or DOHAD paradigm, originally called early origins hypothesis or the Barker hypothesis, and that's David Barker shown there in the right. And then so through a wide range of human cohort, natural experiment, and experimental animal model studies, the DOHAD paradigm has shown us how prenatal and early postnatal environments during sensitive periods of rapid growth and development have lasting effects on cardiovascular and metabolic systems with implications for disease risk in adulthood. So what about the immune system? Most of the DOHAD literature has focused on cardiovascular pathways and metabolic pathways and cardiovascular disease and metabolic disease outcomes. Not much attention has been paid to the immune system. But as I said earlier, inflammation is a really important part of the immune system, and infancy is actually a really critical period of immune development. So if we just look at sort of the natural history of what we know about normal immune development, this is what it looks like. The red line shows you where you end up as an adult. The top graph is these are sort of enumerative measures, numbers of different types of cells, concentrations of immunoglobulins. Down here we have functional measures, how likely these cells are to proliferate once they're stimulated, which is an important part of the immune response. And basically the takeaway here is look what's going on in infancy, right? So the immune system is on hyperdrive in infancy and early childhood, and then it slows down and gets to adult levels, basically through adolescence. So it's kind of analogous to the brain. There's just a lot of activity early in life, and this is suggestive of a system that is primed to learn from its environment or engage inputs from the environment to drive its development and ultimately drive its function. So just looking at this, one would say, well, if we want to understand how this system works here, we need to pay attention to what's happening here, just based on these data alone. So our first effort at this was to look at birth weight. So we first tested the hypothesis that birth weight might be associated with chronic inflammation in young adulthood in the Cebu cohort. So we're drawing on the Barker hypothesis, which focuses a lot on birth weight as a readily available yet imperfect proxy measure of prenatal nutrient transfer that's consistently associated with cardiovascular and metabolic disease risk in hundreds, if not thousands, of studies at this point. So that seemed like a good place to start. We have information on birth weight, we have CRP in young adulthood, and here's what we found. As birth weight increases, the probability of having high CRP goes down. So, this is an observational birth cohort, so there are obviously limitations for causal inference there, but it's consistent with the larger literature suggesting that higher birth weight is associated with better cardiovascular outcomes. And this was important in that context in suggesting that inflammation might be part of that pathway, whereas the research previously had focused on blood pressure, lipids, glucose metabolism, things like that. So again, with respect to this causal inference, we wanted to replicate this finding in a different cohort with some opportunities for natural experiments. So we turned to AdHealth, and we looked at weight for data when participants were 24, 32. I assume pretty much everyone here is familiar with the AdHealth data set, but nationally representative, began with the recruitment of effectively 20,000 kids and adolescents. It's followed them through. There's a nice parent module, which has information looking back, parents reporting on birth weight and other aspects of the early life environments. And then for my purposes here, there's the overrepresentation of siblings. And so we could do sibling fixed effects models to control for shared aspects of family environment, neighborhood factors that could confound associations between birth weight and inflammation later. So this is what we find in the full cohort. As birth weight goes up, levels of CRP come down, a similar pattern of association that we see in the Philippines. And then when we look at the siblings, which there were 714 pairs, we see an even stronger association between birth weight and CRP. And so we sort of replicate the finding in the Philippines, and that points to the importance of birth weight potentially as predicting chronic inflammation later in life. But we wanted to go beyond that and look at breastfeeding as an important part of the postnatal nutritional environment. The DOHAT literature tends not to focus so much on breastfeeding, but there is some literature there. We couldn't test the hypothesis that breastfeeding might be associated with inflammation in the Philippines because everyone is universally breastfed for long periods of time. That's not the case in the United States. Where when the At Health cohort was born, that was actually kind of coming out of the nadir of breastfeeding in the United States. Now we're doing a little bit better, but the current recommendation is 6 months of exclusive breastfeeding. Less than 1 in 7 babies gets there at this point in time. And breastfeeding is interesting because there are good reasons to think that breast milk in particular has a lot of microbial, immunological, and nutritive properties that could plausibly affect inflammation later in life, particularly during that critical period of development in infancy. All right, so in the At Health cohort, we have information on breastfeeding durations. It's not super granular. It's mom's report back in time. So there's going to be a fair amount of measurement error there. But nonetheless, we find that after 3 months of age, so any kid as a young adult who was breastfed as reported by his or her mom for 3 months or longer has significantly reduced CRP as a young adult. And when we look at the SIBs, of which relatively few are discordant, so you can imagine that a family that can support breastfeeding will probably do that for all children. So there's some caveats here as to why these kids are discordant. And we do our best with that, but we see a really big difference around this 3-month threshold that the kids who are breastfed, a sibling breastfed for 3 months or longer compared to one that wasn't breastfed as long, have really different CRP concentrations as young adults. All right, so I want to just take a quick aside here on and make a point about effect sizes. These are the best case clinical trials, gold standard clinical trials for reducing inflammation using statins and some other anti-inflammatories. And what you see in these prospective epidemiologic RCTs, that with the intervention, with the pharmacological intervention, you can get about a 15–18% reduction in CRP in adults. What we find in our At Health cohort, and these are using the more conservative coefficients on the full cohort, not the siblings, that a pound of birth weight, which is a lot, like to give get an extra pound of baby, reduces your CRP 5%, but breastfeeding 3 months or longer has a 20–30% effect on CRP. So this is something that I'm very interested in. And if any of you are also interested in this question and have some ideas for data sets or opportunities to further investigate that, I'd be very interested in talking to you. Breastfeeding is hopelessly confounded in the United States. We do a horrible job of supporting moms and families with respect to breastfeeding. We're like the only country that doesn't have a reasonable family leave policy, paid family leave policy, and that's directly related to breastfeeding initiation and duration. So I'm interested in doing research that might help us understand better the long-term effects of breastfeeding in multiple health dimensions beyond just inflammation. All right, so we're talking about the immune system here. So we've been talking about nutritional aspects of the nutritional environment that may matter to inflammation, but the immune system is really largely about preventing infections from taking over our bodies. And so microbial inputs are an important part of what the immune system is all about. And many of you are probably familiar with this idea that actually some germs, exposure to germs, are good for us and actually necessary for our immune system to drive its development. And we have this notion of the hygiene hypothesis, which has been around for a little bit more than 25 years, showing that kids in particular who have older siblings, were exposed to other kids through daycare, live rurally on farms, have higher levels of infections or microbial exposures in infancy and early childhood, they're less likely to develop allergies and asthma as children. And there's a lot of immunology around that, but it's all focused around childhood immunology and atopic, asthmatic, and other allergic diseases. The implications for inflammation, not known. So we decided to look at this, and this is one of the opportunities we have, the Cebu cohort, which was in the midst of classic epidemiologic transition in the 70s through the 90s, where rates of infectious diseases were coming down, rates of chronic degenerative diseases starting to go up, the study started in 1983. And at that point in time, about half the homes had electricity. More than 3/4 of the homes were relying on water from an open source, like a well or a stream. Less than half had a flush toilet on the premises. And more than half of the families had animals, domestic animals, goats, pigs, chickens, dogs, in the house, under the house, or around the house. So a much richer, more diverse microbial experience for many of the kids there than certainly what I had growing up. But an interesting thing about Cebu is there's also a wide range of variability. So there are very affluent suburban communities that were part of the study, as well as squatter settlements and everything in between. So a pretty wide range of exposure in this domain, as well as others. All right, so what do we find here? So we looked at a measure of infectious diarrhea in the first 2 years of life. This was an interviewer went into the home every 2 months and interviewed the mom, weighed the kid, asked about symptoms of infections or the preceding week. And so it's not a complete census, but over 24 bi-monthly interviews, it's pretty good relative assessment of who had higher levels of infection versus others. Kids who had more episodes of documented infectious diarrhea in infancy have lower probability of high CRP as a young adult. We also have a measure, it's called frequency of fecal exposure. So this is when the interviewer went into the home and observed that a child was in contact with those domestic animals, or had the opportunity to be in the same space. So it's a proxy measure of exposure to animal feces, basically. Kids who are more frequently in that sort of situation in infancy have lower CRP as a young adult. And then we also have season of birth. So the Philippines is a highly seasonal tropical environment. And if you happen to be born in the dry season, postnatally after birth, you were exposed to more infections. You had a higher rate of infectious disease in infants. So we use this as a proxy for infectious exposures in the first year of life. And those kids with more infections have lower CRP as young adults. So we have 3 different measures here. Sorry. All of which tell us something about the microbial infectious environment in infants. And I wouldn't spend too much time interpreting any one of them, but all 3 sort of different sources of data, different measures converge on the same conclusion that higher rates of microbial exposure in infancy reduce levels of chronic inflammation later in life. So this is kind of an extension of this hygiene hypothesis. So let me interpret for you what I think might be going on here. So if you live in a high microbial environment in infancy, when your system is on overdrive and sort of primed to engage with the environment and develop, then you're getting more sort of opportunities to activate the system. That's what these red arrows are, sort of microbial inputs. They aren't necessarily pathogenic. It's not necessarily the flu or polio. It can be relatively benign microbial, but your body still has to deal with that. So it activates the system, and then you resolve that, but that happens repeatedly. And then as an adult, to the extent that these experiences have a lasting impact and are sort of remembered by the body, you have a more efficient, tightly regulated inflammatory system that's less likely to become chronically inflamed. If you grow up in a relatively impoverished microbial environment, you don't have as much exposure during that priming period of immune development. And then the system just doesn't really know how to deal the same kinds of pro-inflammatory stimuli later in life. So it leads to chronic levels of activation, or when the system gets activated, it becomes overblown or can't resolve a response once it's put in motion. And so that, I think, might explain this pattern of difference, right? So here's the chronic low-grade inflammation model as based on research in the US with multiple measures of CRP. Here's what we see in Ecuador. So qualitatively different patterns of production of CRP and inflammation over time. All right, and so for the broader purposes of the argument right now about why we shouldn't start our aging studies at age 50, we have a lot of evidence to suggest now that aspects of the microbial environment, and just an example I've given you, sanitation, hygiene, infectious exposure, aspects of pre and postnatal nutrition, as indicated by birth weight and breastfeeding. So large and emerging literature on adverse childhood events and how they affect inflammation later in life, which I'm not talking about. All these things, all these early life exposures, which happen well before 50, matter to inflammation. So if we start our studies of aging at 50, then we're going to miss the path of developmental plasticity, ecological contingency. They're really important design features of the immune system, and then exposures during this period have lasting effects on the regulation of inflammation, and that might matter for the associations between inflammation, aging, and disease. All right, last assumption. So I already shown you how Time Magazine branded inflammation the secret killer, but it's not just the media that's sort of going overboard here. So this is a paper that's reviewing transcription factors. It's from Nature Immunology, one of the top immunology journals, and they're reviewing transcription factors that control immune responses. And the authors refer to these basic intracellular regulatory pathways as being associated with diseases ranging from inflammation to cancer. So inflammation is like the same as cancer here. We have the inflammaging paradigm, which is based a lot on research with fruit flies and animal models, showing how oxidative stress and other inflammatory processes cause damage to cells, and that cellular debris and damage in turn upregulates inflammatory processes. You can get in this feed-forward cycle of inflammation causing damage, and then that damage eliciting a cellular level and then systemic inflammatory response, and that can drive functional decline and increased risk for disease. That's the inflammaging paradigm. So inflammation is basically a driver of disease and aging in this paradigm. And then many of you are probably familiar with the cohort morbidity hypothesis that was proposed by Eileen Crimmins and Tucker Finch, which suggests that historical increases in human life expectancy are due in part to reductions in infectious disease. And it's not just the increased survival through childhood, it's that reductions in overall lifetime burdens of infectious disease reduced overall lifetime exposure to inflammation, which then reduce risk of cardiovascular disease and other diseases of aging, and led to increases of life expectancy. So inflammation is part of that sort of story. Okay, so what's wrong with that? Why should we not, or why can't we assume that inflammation is pathological? Well, at first, it ignores the fact that inflammation is critical to survival in terms of protecting us against infectious diseases, involved in wound healing and tissue repair and these basic physiological processes like brain growth and development, like I said earlier. But also, and I think more, I don't know if it's perniciously, but importantly, it encourages us to make some relatively simplistic assumptions that may structure our research in this area and potentially counterproductive. So I want to give you an example here. I'm going to pick on Mike Gervin and his colleagues, and I know all these people and they're great. And then I could have picked other studies, but there's a reason why I picked this one. So what Gervin and colleagues did is they did a study, you can't quite see the title, but it's called Aging and Inflammation in Two Epidemiological Worlds. So they're comparing levels of inflammation in the US with levels of inflammation among the Chimani, which is a lowland Amerindian group in the Bolivian Amazon basin. And so that's a place actually that I've worked. I took this picture. They very similar to the Shuar, high levels of infectious disease, particularly early in life, live in small villages on a system of rivers with small scale horticulture, hunting and gathering, things like that. And so what Gervin et al. Did is they collected blood samples from kids as young as 5 and then adults 65 and up. And then they compared that to NHANES data and then just plotted it here. So you can see this is the prevalence of high CRP. What proportion of these people in this age bin had CRP above that high risk threshold? And at each point, there was significantly higher proportion of Chimani adults with high risk CRP than in the United States. Okay, so that's the paper. That's the finding. And based on that, they say that based on the association of CRP elevations with future cardiovascular events in populations with lower mortality infections, we hypothesize that premature cardiovascular disease is a factor in the higher adult mortality of the Chimani. So I think this is a good example of how the chronic low-grade inflammation paradigm can encourage us to make some relatively simplistic assumptions here. So the assumption here is that one measure of CRP gives you a measure of overall chronic level of inflammation, and that higher level of inflammation is basically toxic that increases your cardiovascular risk, which leads to high mortality. So a pretty linear set of associations that may not be warranted. So let's take a look at that. We have to be careful, I think, about assuming that inflammation is indeed always synonymous with pathology when we know that it actually does a lot of good things for our body. And we see in those Ecuador data that actually maybe having an acute response that goes up, then being able to resolve might be a good, a good regulatory phenotype to have in play. But how can we study inflammation and gain insight into those dynamics if we're just measuring baseline samples with CRP at sort of 1 or 2 points at a time? So to try to get around that, we designed a study with our cohort in the Philippines, where we use the CRP response to vaccination as an in vivo model for the regulation of inflammation. inflammation. So just instead of just measuring CRP at one point in time, we activated the inflammatory response with a mild pro-inflammatory stimulus, which is the flu vaccine. So this allows us to look at how some people might have a more sort of tightly regulated response, more diffusely regulated, and maybe that matters for the association between inflammation and disease. We did this with 934 women, mean age of 55 years, gave them the vaccine. We used dry blood spot sampling to get a baseline sample right before the vaccination, and then a follow-up sample 3 days later. We wanted to do something more intensive and over longer periods of time, but it was a compromise with our colleagues in Cebu, so we could only do one follow-up. And on average, what you see at baseline, the median CRP is 1.2 milligrams per liter, and then the median at day 3 is about 1.5. So about a 0.3 milligram per liter on average increase in CRP in response to the vaccination. So sort of validating this as a mild pro-inflammatory stimulus, but obviously there's a lot of variability there. So this is the median response. There were a lot of women who didn't respond at all, and then there were a lot who responded really robustly. So that's the interesting information that we want to test, we want to interrogate here. So what does that mean? So how do I interpret this clinically, how this might matter to the development of disease and age-related chronic degenerative diseases? We did a follow-up 3 years later where we measured ankle brachial index. It's you basically measure blood pressure in the arms and in the legs, and you calculate a ratio, and that gives you a sense of peripheral artery disease, which is highly correlated with cardiovascular disease risk in general. So this is actually a diagnostic procedure that we were able to implement with local cardiologists. And we simply wanted to test whether baseline inflammation and or the CRP response to vaccination were predictive of subsequent cardiovascular disease risk. And so these are what the data look like. So we just look at chronic inflammation at baseline, higher CRP, and cardiovascular risk. So ABI is a higher score is better. So individuals who had lower CRP at baseline had lower CVD risk, which is what we expect to see. But then when we look at the response to vaccination, so a higher magnitude of response positively associated with ankle brachial index, lower CVD risk. And we put both those things in the model, the baseline CRP drops out. So women on average who responded really strongly to the vaccine had lower baseline levels of CRP, and that response was strongly protective with respect to future CVD risk. So a higher CRP, in this case, was associated with lower CVD risk. So we're interpreting this as, well, maybe if you have this really robust system that's not really turned on except when it's needed, and then you activate it and it responds really robustly to deal with that challenge, you have a tightly regulated system that doesn't contribute to cardiovascular disease or other diseases of aging. Whereas if you didn't respond, you sort of high baseline levels of systemic activity, that's not good. But that might actually contribute to the development of disease. All right, so coming back to Gerben and their study. So they proposed that high inflammation is associated with high cardiovascular risk and high mortality. Our study suggested that we can't assume that that's the case. And to their credit, 1 year later, they published a study that said the same thing. So they took their sample and they looked at cardiovascular risk factors among the adults, and they found no association between baseline inflammation and subsequent cardiovascular disease risk. And there are now a handful of studies that are starting to sort of break the link between inflammation and diseases of aging in non-Western, higher infectious disease environments, including Ghana, Malawi, Philippines, Bolivia, Ecuador. So it's interesting. All right. So what does this all mean? I'm gonna wrap up here. We don't have time for conversation. There's a large and growing body of research in industrialized settings that does tell us that chronic inflammation is a cause and consequence of aging, and that it contributes to chronic degenerative diseases and earlier death. So inflammation is pathological. There does seem to be, with a lot of studies and various cohort studies, degenerative diseases, they are related to aging, and chronic inflammation is part of this sort of feed forward cycle. But does it have to be that way? What are we missing when we conflate inflammation and pathology? If we construct chronic low-grade inflammation as an inexorable component of age-related disease, I think we overlook what might be possible. we have substantial evidence now from diverse settings globally to suggest to us that chronic inflammation is not necessarily normal. We don't want to assume that that's universal or normal, and it's not always associated with chronic degenerative diseases. So if we recognize that chronic inflammation is not always part of aging, then the question becomes, what determines an individual's inflammatory phenotype? So getting Amelia to your question earlier, what circumstances lead to a more tightly regulated system? One versus one that's always on or diffusely regulated that can't turn itself off. We have evidence to suggest that aspects of pre and postnatal nutrition, aspects of psychosocial stressors over development, which I didn't talk about, but I can be happy to answer questions about, and then microbial exposures. These are all important for shaping how your body regulates inflammation. And that might be sort of a bifurcation point for how your body responds to inflammatory stressors later in life. Does it get caught up in a cycle, a sort of downward spiral of inflammation and aging, or can you break the link between inflammation, chronic degenerative diseases, and aging? All right, so where do we go from here? I think we need to complement current, excellent epidemiologic research in places like the US with studies conducted in more divergent ecological and epidemiological settings around the world. Methods are a challenge, but we've, I think, solved a lot of those problems. I'd be happy to talk about that. We need to evaluate how associations among inflammation, aging, and disease are shaped by environments early in life during sensitive periods of development. So early aspects of the environment are highly relevant to how we understand aging later in life. And we also need to consider more dynamic measures of inflammatory activity. Single baseline measures of CRP, other inflammatory cytokines are very limiting. They encourage us to see something as chronic when in fact it's very dynamic. And we need to have more dynamic measures if we really want to generate insight into these processes. All right, I'd like to thank our funders for all these various studies, my colleagues, and thank you very much for the invitation.
Thank you, Thom. Now, questions?
So I agree very much that, aging probably starts at conception. But I wonder, what do you think about how to undertake research? So we can start research today with a sample that was just conceived. And in 60 or 80 years, when I don't think I'll be around, I don't think anyone in the room will be around. Someone can analyze those data. But I guess, the question is, how good, or do you have a view on this? How good is the alternative of effectively patching together different life cycle segments? Using Ad Health for part of the HRS for another part, if we're going to be US focused, et cetera.
Yeah, I mean, I think we just have to do the best we can, right? But I think that's a real limitation. In terms of where you started with, aging, it began at conception. I think we are generating knowledge and tools that allow us to study age-related processes much earlier in life course than we have been able to do previously. And part of that's inflammation, but there's a whole expanding literature on measures of epigenetic age or biological aging. So looking at our genome and the marks, the chemical marks on it, the epigenetic marks, DNA methylation in particular. So there are many of these epigenetic clocks that get at the pace of biological aging, many of which are actually starting to be used in adolescence and in childhood. And it's not entirely clear, if you are older biologically as a 6-year-old, whether that's a good thing or not, but it's an empirically observable phenomenon at this point. So I think there are some interesting opportunities there. And aging, I think aging is often conceptualized as your body grows, develops, and it reaches a peak and then it declines, and you're kind of trying to fight that off. But the body is constantly dynamically reconstructing itself. So there are reparative, restorative processes in play in childhood and adolescence that we can measure as well. So I think there are opportunities to have broader life course approach. I don't know how to fix the cohort problem without a sort of time machine situation, but do the best we can.
Yeah, you mentioned this in passing. There's a relationship between inflammation and depression, other psychiatric disorders. So I'm curious if the same sort of analogy or metaphor framework can be applied. That do people have a psychological system that's more efficient, dynamic, adaptive to their present circumstance as an adult if they're exposed to more fecal matter as a child, or
Yeah.
whatever, however you want to think about that
Yeah,
process.
I think I'll give you two examples, and I think the answer is yes. And I think that's a really interesting and promising area of research. So in Cebu, we have measures of depressive symptoms in the kids and the moms. It's like, it's an adaptation of standard instruments from the United States, so there's some caveats there. But we find no association between our markers of inflammation and depressive symptoms there. And then when in the moms, and then when we look at it in the kids, we see a hint of it, that higher levels of depression or higher reports of stress are positively associated with CRP in the kids who are raised in the most hygienic circumstances. And the kids who are raised in the more typical microbially diverse situation, it breaks down. There was a recent study, I'd be happy to send you the link or the paper, where the investigators implemented the Trier social stress test, which is a social evaluative stressor. It increases cortisol, it increases inflammatory cytokines over the course of hours, and then they come back down. They did this with two groups of young adults, or probably college students, kids who were raised on a farm and kids who were not raised on a farm. And kids raised on the farm did not show the increase in inflammation, whereas the other kids did. So I think there are lots of ramifications of the story. So if it's true that a rich microbial environment qualitatively shapes your inflammatory phenotype in a way that it reacts to a threat and then sort of shuts itself down and doesn't contribute to pathological processes as we age, that matters to depression and lots of psychiatric disorders. It matters to dementia, matters to cardiovascular disease, it matters to diabetes. It could matter to everything that has an inflammatory component, which seems to be everything. So.
Michelle?
Thanks, Thom. Really great fascinating talk. Can you tell us a little bit more about the pathways that explain the connection between breastfeeding and inflammation? Is it a nutrition story, or is it more about acquired or passive immunity? That was my first question. Second question, when you compare US data with Cebu data or other countries, are you thinking at all about mortality selection that potentially could affect?
Yeah, I'll start with the second one first because I have a less satisfying answer, but we're aware of that. And in Cebu, the mortality selection issue is, I think, not as big a deal as we might have anticipated. Mortality rates in infancy were relatively low, I think lower than Barry Popkin and other folks anticipated early on that it would be. We're more concerned about migration selection because we started with 3,300 so kids, and now we're down to half that. And there's a lot of out migration from Cebu, and we haven't had the resources to track. So we do worry about that. But I think most of the bias is probably away from the things we're finding. With respect to breastfeeding, breastfeeding is a nutritional intervention, and there are a lot of prebiotic and nutritive substances that affect immune development, and particularly the colonization of the gut microbiota, which can have lasting effects. So that's one mechanism. There are a lot of cellular and soluble immune factors in breast milk, which help the infant's immune system develop more quickly and buffer it against infectious exposures. So that's one of the reasons why breastfeeding protects against allergies, asthma, things like that. What we find here, when we do mediation analyses with what we have available, which is measures of body fat, that breastfeeding is associated with lower waist circumference as an adult, and that accounts for about half the association between breastfeeding duration and CRP in young adulthood. So sort of body fat accumulation is a big part of that story. Yeah.
I'm going to follow up a little bit on the mortality question about this early exposure to infectious diseases and CRP and lower CRP. I mean, the diseases that you mentioned, like diarrhea or something, they also kill you.
Right.
So, so in developing, is there a trade-off here? Or how does, does it produce selection, or is there a right level of exposure?
Yeah, I think there probably is. I don't think we know what it is. I think if we're talking about the priming of the immune system and helping it develop to be more tightly regulated, the exposures that are necessary are not the ones that will kill you. It's not the pathogenic bacteria and viruses. So, vaccination, campaigns to reduce exposure to cholera, I think those probably are good because they protect you from death against infectious disease, but they don't necessarily throw the baby out with the bathwater. There are millions of benign and beneficial bacteria in soil and food and water that we've scrubbed out of our supply chains here, but a lot of people and historically have been exposed to. And those are the things that probably are really important for priming the immune system and helping in development. So they don't lead to clinical disease, but you tend to remove them from your environment in the same as part of the process for removing exposure to cholera and polio and those things as well, right? So that we end up throwing the baby out with the bathwater. So what can we do about that? This was an interesting study in Finland where they, at a daycare setting, they took what they call a forest floor. So they literally took dirt from the forest and they put it in the play area of the daycare. And then they measured the gut microbiota of the kids and then some immune factors related in blood to inflammation, like a month after the intervention, compared to kids playing on concrete, kids on the forest floor. Those kids obviously had a much richer gut microbiota and lower inflammatory profile, just even after a couple of months. So it could be things like that. I know some folks are interested in probiotics and prebiotics as a way to get some exposure to the good bacteria without exposure to the bad ones, but those don't seem to be very effective. But I think ultimately it's going to have to be some environmental manipulation or some pharmacological one.
Interesting. So.
I mean, I also can tell you what we do in my house. I mean, I have two kids and I don't know if it makes a difference because they actually have allergies, but we don't, we encourage them to play in the dirt. We don't, when we buy vegetables that are organic from like a farm market or something, we kind of wash them, but we don't scrub them and sanitize them. That's an effective way to get some access to healthy bacteria.
So one final question, yes.
I want to ask about sedentary lifestyles because it could be like something that causes the internal or the in-unit correlation in the US and developed world and not in the developing world. I imagine everyone's more moving around a bit more.
Yeah.
But it didn't come up in the talk at all. So
Yeah, no, that's a very good point. Cebu has actually become quite sedentary. Now it didn't used to be, but just sort of various economic transitions, and they have a rising rate of overweight obesity in part because of that. I think physical activity matters for a couple of ways. One is if you're more physically active, you're less likely to accumulate body fat, and that will reduce the burden of inflammation. But physical activity in its own right actually can help your body manage inflammation. So exercise is initially pro-inflammatory. It activates some inflammatory pathways, but in a good way that leads to repair of damaged muscle, and then the system gets turned down. So it actually can be a way to sort of activate and train the system to go up and down. So there's a lot of emphasis in exercise science talking about the beneficial anti-inflammatory effects of exercise. So that could be part of the story as well.
Okay, thank you,
All right,
Thom,
thank you.
very much.
That's it. I know, but well, the story, I started work my class when I was here. That's like.
Studies in Mexico.
But yeah. Anyway, the other.
Anyway,
I appreciate the invitation.
So it's great to see.
Yeah, I'm like, yeah, but.
Mexico, where you also made.
Yeah, I think that's interesting. The issue of exposure, we all know, I do, coming from Argentina, I do see that some of the disease in the US is, lack of exposure to something. But at the same time, there is that, like, you don't want to be exposed
Right,
too
no,
much.
exactly. You don't want too much.
Very
Hey,
good.
hey,
See you.
yeah, I'll see you shortly. Yeah,
3:30.
yeah, sounds good.
Yeah, you don't want to be exposed too
Right,
much.
I mean, no, exactly. So, yeah, when we first published the paper linking microbial exposure to lower CRP in the Philippines, that was the first time that had been shown. So other people have shown it since, and it got picked up in the media. It was on CNN, and I was doing a lot of interviews, and people were saying, "So does that mean I shouldn't vaccinate my kids?"
I didn't think about it, but now you mentioned it.
They
because
should get.
That's what they're
flu.
saying, yeah.
Yeah, and I said, no, absolutely. And like I said to you, there are bugs that will kill you. You don't want those. And they're not the ones that our
I
bodies.
don't say that diarrhea is a good thing.
Not saying that. Yeah.
Anyway, so let's go demonstrate. Yes, I'm gonna do lunch and.
All right.
Yeah.
How you say? Hi, nice to meet you. All right. Yeah. All right, where are we going? The Spiral. Thanks, Emilio. Take care. Bye. I was hoping I could go over this time. Very interesting. Thank you.